Your body replaces roughly 330 billion cells every single day through a tightly regulated process called cell division. Normally this process is controlled by a precise set of molecular brakes and accelerators encoded in your DNA. Cancer begins when that control system breaks down, not because an alien force invades your body, but because your own cells accumulate genetic mutations that cause them to ignore the signals telling them to stop dividing. Approximately 1.9 million new cancer diagnoses are made in the United States each year, and yet the vast majority of cancers are not inherited. Only about 5 to 10% are driven by hereditary mutations. The rest arise from accumulated errors in DNA replication, from environmental exposures, from viral infections, or simply from the random copying mistakes that happen every time a cell divides over a lifetime. At the molecular level, cancer is fundamentally a disease of the genome. Two categories of genes are at the center of the story. Proto-oncogenes are normal genes that, when mutated into their cancer-driving form called oncogenes, send constant go-go-go signals to a cell regardless of external cues. Classic examples include RAS, mutated in roughly 30% of all human cancers, and MYC, which drives uncontrolled gene expression. On the other side are tumor suppressor genes, whose job is to put the brakes on cell growth. The most famous is p53, sometimes called the guardian of the genome, which normally detects DNA damage and either triggers repair or orders the cell to self-destruct through a process called apoptosis. When p53 is mutated, and it is mutated in over half of all human cancers, that guardian is silenced. BRCA1 and BRCA2 are tumor suppressors important in DNA repair. Their loss dramatically elevates breast and ovarian cancer risk. Cancer researchers Douglas Hanahan and Robert Weinberg described what they called the hallmarks of cancer, a framework of biological capabilities that virtually all cancers must acquire. These include sustained proliferative signaling, meaning the cell tells itself to keep dividing, evading growth suppressors, meaning it ignores the stop signals, resisting cell death, meaning apoptosis is switched off, enabling replicative immortality, meaning normal cells can only divide a finite number of times but cancer cells bypass that limit by maintaining their telomeres, inducing angiogenesis, meaning the tumor grows its own blood supply by releasing signals like VEGF that recruit new blood vessels, and activating invasion and metastasis, meaning cells acquire the ability to break away from the primary tumor, enter the bloodstream or lymph system, and seed new tumors in distant organs. No single mutation produces all these capabilities. Cancer is a multi-step evolutionary process that unfolds over years or decades. One of the most counterintuitive facts about cancer metabolism was described by Otto Warburg in the 1920s and is now known as the Warburg effect. Normal cells generate energy very efficiently through a process called oxidative phosphorylation that uses oxygen in the mitochondria. Cancer cells, even when oxygen is plentiful, largely switch to a less efficient pathway called aerobic glycolysis, breaking down glucose without fully using oxygen and producing lactic acid as a byproduct. This seems wasteful, but it actually serves the cancer cell's needs. Glycolysis is much faster and produces the molecular building blocks the rapidly dividing cell needs for new membranes, proteins, and nucleic acids. PET scans exploit this glucose hunger by tagging glucose with a radioactive marker that cancer cells greedily absorb, lighting up tumors on the scan. Metastasis, the spread of cancer from its original site to distant organs, is responsible for about 90% of cancer deaths. The process involves multiple steps. First, cancer cells at the primary tumor undergo what is called an epithelial to mesenchymal transition, changing their shape and surface proteins to become more motile and invasive. They then digest through the extracellular matrix using enzymes called matrix metalloproteinases, enter a blood vessel or lymphatic channel, survive in circulation despite lacking the adhesion signals normally required for cell survival, and then extravasate, exit through the vessel wall at a distant site. Finally, those cells must establish themselves in a completely different tissue environment, which only a small fraction of circulating tumor cells successfully achieve. The organ specificity of metastasis, why prostate cancer preferentially spreads to bone, or lung cancer to the brain, is an active area of research involving the concept of a premetastatic niche. Modern cancer treatment has been transformed by the understanding of cancer's molecular biology. Targeted therapies block specific mutant proteins. Imatinib, for example, targets the BCR-ABL fusion protein in chronic myeloid leukemia with extraordinary precision. Immunotherapy, particularly checkpoint inhibitors that block proteins like PD-1 and CTLA-4, releases the immune system's existing ability to recognize and kill cancer cells that had learned to hide from it. CAR-T cell therapy engineers a patient's own immune cells to recognize and attack their specific tumor. The field has moved from a mostly cytotoxic approach, kill everything that divides fast, toward a molecularly precise approach that exploits the specific vulnerabilities of each patient's cancer. Understanding how cancer works mechanistically is what makes these interventions possible, and it is why cancer biology remains one of the most active frontiers in all of science.